And you asked me what's the latest thing, because the NMN research that we did, we're still doing a bit of it. We just are about to submit a paper showing that NMN extends lifespan in mice, um, especially female mice. And so that's coming. Most of my research now has been directed at understanding the software of the body beyond the sirtuins. What else is involved in causing aging to occur, and how do you reset that? And we had a paper published in Cell in January last year that showed evidence that changes to the epigenome can drive aging. So we made a mouse. It took us 13 years to do this research. Made a mouse where we could disrupt the epigenome. We did this by creating cuts to the DNA when the mice were young. As far as we can tell, that didn't disrupt the DNA. The DNA just got put back together quite easily. But the epigenome got disrupted. And we know that damage to cells disrupts the epigenome over time. And we'll get to this, but stopping damage to your cells is one good way to slow down aging, because it slows down epigenetic changes, slows down the software corruption. But this paper, what it showed was that we could drive aging forwards by disrupting the epigenome. And we could also use 3 of the Yamanaka factors. There are normally 4 used, but a particular subset of 3 was able to safely reverse aging in those mice. And how that works, we don't know. We think there are little flags on the genes that tell the cell this gene needs to be reset by 2-fold in a negative direction, or another gene would be 5-fold in a positive direction. There's some information in there. We're looking for it. We have some breakthroughs just last week that I can't yet talk about. I think we may have found the information where and how it's stored that tells the cell where to go and what to do. And it's a whole new biology. So I'm crossing my fingers for my students that we're on the right track. What the Yamanaka factors are, they're not that mysterious. These are proteins that engage DNA. They find a sequence of letters in DNA. They're called transcription factors. And they find them, they stick to the DNA, and then they set off the activation of other genes. So these are genes that regulate other genes. That's all they are. But they're like a domino. Those 3 dominoes get set, and then the rest follows. And then the complexity ensues. But there's got to, in my view, there's got to be a program in the form of software, one-dimensional software, just like DNA, but probably not DNA, that records the youthful epigenome and allows the cell to re-engage and create these original structures of folding of the DNA, so the cell can not just behave like it's young, but literally be young again. The answer is yes. Somehow the Yamanaka factors know how to reset different types of cells. We haven't yet seen a cell type that does not respond to the reprogramming, and each cell type is different. Of course, a brain cell is very different in terms of the packaging that it needs compared to a muscle cell. And so, yeah, there is a culture within the cell. Perhaps it's this system that we're trying to figure out, which is this backup copy that is specific to each cell. And so the cell individually, they know what they need to go back to, and it's perhaps different literally for every cell in the body, the pattern that needs to go back to. And we don't even know yet how and when that is laid down. Is it when the baby is born? Is it when you're a teenager? Is it different for different organs? Does your liver become set in youth at 2 and your brain at 20? These are really interesting questions that we want to address. And we published a paper, when was it? July last year, the first proof of concept study that we could find chemical cocktails that reversed the age of cells and restored that epigenome. And we've come a long way since then. It's not published yet, but we are down to some really exciting chemicals that could theoretically be put into a drink and consumed, that may rejuvenate the body, improve memory, for example. I think, don't get me wrong, I'm not going to be putting this into a drink just this year. But I think if the animal studies work out and we do some human studies, then that's, it's a goal worthy of my attention. There's cocktails, so there isn't just one answer. But we're at a point where we now have it down to one chemical that can reverse the age, ostensibly reverse the age of cells in the dish, can treat human cells, make them younger, make them grow, even if they stop dividing, senescent cells. And barring any safety issues, which of course you can't ignore, um, it could be made available. So right now, could you go out and buy some of these? Well, I do know that some people have looked at our publication from July and have started taking those chemicals or surrogates of those. But they're doctors, you need a prescription, so it's not that simple. You can't just go out and buy it. And it's not just a vitamin, it's a bit more complicated than that. But just trust that I'm now laser focused on making this come true if it's possible, if it's safe. It's not here yet, but it will be if I can do anything about it. We haven't put them side by side, but I don't think anything that is out there compares to this. We don't see the usual suspects, rapamycin, NMN, doing this reset. They might assist in the process, but these chemicals are in a different league of their own. And so I think this is probably why people are skeptical about my use of the term age reversal, because they're not seeing the results that I see in my lab. And we clearly see that aging is reversible. And these new discoveries are really powerful, more powerful than anything I've seen before. It's different. What worked in the mice was we stabilized their DNA, and that's the main problem with infertility in females. The eggs trash their chromosomes, and there was a protein, one of the sirtuins, that actually was the problem. And by raising NAD, we could make sure the chromosomes were nicely packaged in the egg. An old egg, by the way, the chromosomes are all torn apart. It's quite terrible. There's no way you're going to produce an offspring for one of those. But it's different. It's different. This new age reset isn't just for eggs. It seems to work on every cell type. And you don't just go back a little bit. You seem to go back a lot, 50%, 75% in age, and reset the pattern of what we call gene expression. And what's interesting is it's not just a temporary reset. We've co-published a paper recently that when we essentially cure glaucoma in a mouse and turn off the Yamanaka factors, the effect continues, right? It's not that you need to keep the factors on. It's truly a reset, and then the mouse gets old again, and then you just can keep resetting. We know we can reset the body at least twice. I'm not sure there's actually a limit to it, other than in these experiments, the mice got old and died, but their eyes were beautiful at the end of the experiment. But yeah, so it's, I'm looking forward to us getting better at reprogramming the body of mice so we can make them live longer. There was a recent study that was published, not from my lab, but a group that used our system, and they gave it to old mice. They put the Yamanaka virus into the vein instead of the eye, and those mice were very old, and their remaining lifespan was doubled, 109% extra life.
Wow.
Which, is a good beginning, but I would love to be able to make a mouse that could live for, 2-3 times. But the technology's not really there yet. The biggest obstacle is getting the virus into all the cells evenly. And viruses like to pile up in the liver and a few other places. And so what people are doing, not me, but people who have the skill in viruses, they're engineering viruses and even other types of delivery vehicles like nanoparticles that can evenly spread across the body and deliver genes.